Well, it's of course a pleasure to talk today here, because I think it's a wonderful day. We are all very happy and proud to have this new imaging center. And I want to show you, unfortunately, because of time, highly selected a few of the work, and unfortunately also biased more to my group, but I also some work of Elias' group, so a little bit broader. But I want to apologize for those who do not see their work, had to select, and I still have twice as many slides as I should have, so I count on Elia to stop me at some point. So the major idea I want to convey in my talk is actually what Jack Gallant and Camille Goebel have alluded to already, namely that I think that an important step towards the future is to go from what we call the system level or macroscopic scale, like when we have these localized brain areas and so on, and then to move on to what we call the mesoscopic scale. And in this nice slide from Allard, I want to highlight exactly that point, that for both for anatomical data, for structural data, as well as for functional data, we try in our center, and of course in many other centers, to push the limits so that we really reach into the mesoscopic scale. And I have learned this the hard way, to use the term mesoscopic scale, because when I try to publish paper and say columnar, then many people hate this term and say you will not achieve this MRI, and we get a lot of problems there. So mesoscopic scale is now my new term, which I use when I talk about columns and layers. And of course I still will do it, but just that you know, mesoscopic is appearing now more in our submitted papers, because I think it's a bit more easier to convey. The idea is, of course, the one which you, of course, all expect, namely to see both structurally more detail so that we can really separate the layers of the cortex, see connectivity layer specific, see tangentially in the cortex, like different representations of neighboring voxels coding for different features, which are coded by this space. This is then going into the functional domain. And we have heard from Ralf Galuske this morning from the wonderful work Allard is heading here, and with Ralf and many other colleagues, where we do exactly this step into the mesoscopic scale in anatomical world. But I will focus in my talk more on the functional side, because we have heard a bit about this already. And of course, for those who haven't seen the center, that's actually how it looks like. It's actually a three-building complex. So we have the building where the scientists are sitting, using the scanners, locally at least those. Then we have an incubator building here where spin-offs and companies can be housed. And we have, of course, the important building for us, which houses the three scanners, which we now use regularly. And here just some pictures of the siting. So we got the scanners over the last couple of years, and actually including the 9.4 Tesla now, and get also, and I'll show you one image later on, also nice pictures. This is from the 70s. This was the first image where our CEO from Scannexus, Ross McLellan, was the first subject in the scanner, and it's really brilliant data quality. And I have here a slide from Joel Reitler, who gave me that. He looked at the hippocampus, it's a more recently recorded image from the 70s, and it's really, I think, amazing how detailed you see all the folds here in the hippocampus, what quality we can get in a really few hundred microns in vivo here in this case. So it's really impressive what we can achieve with this machine. And as Ralf has already pointed out this morning, and where Allard and I collaborate heavily on, we want to push forward from the macroscopic to the more mesoscopic scale to reach and separate layers and columns from each other and see how far we can come. Of course, this is an empirical question. Whether it's possible, how far it's possible, that we have to find out. That's actually the challenge we have. And I want to add to Ralf's nice presentation an additional work of Matteo Bastiani with Allard and us, which is really impressive in my opinion, because he does something using diffusion imaging, is able to basically separate cortical layers from each other, and can validate this with myelin staining, that you really have the right parcellation of the layers from gray matter diffusion data. So I think that is really exciting. And it's not only possible to separate the cortical layers by this diffusion-weighted technique, but also separate the anatomical areas, like here the motor and the premotor areas, from each other, purely based by kind of going in the depths of the cortex and looking how this kind of diffusion data changes, and when it changes more strongly, that could be a border of an area or across the layers. And I think these techniques, if they work at one point also in vivo, I think would really allow us to kind of get much more neuroanatomically safe grounding for our interpretation of, say, functional data. And of course, we also get nice functional data. It's also a slide from Joel, which basically shows us that if we do things like mapping the phase area here with 3D stimuli, which Joel is using for some phase experiments. And you basically see how beautiful the signal can be on the 7T, right? You get in this little spot here in the phase area quite selective responses to phases and almost nothing to objects. And this, of course, shows as if we would use the machines to do classical experiments, categorical level experiments, we would have a nice time because it's so much nicer now, right? It's strong quality. But if you have better devices, you want to push them and try to find smaller things, smaller differences. And you have the same problems as you had in 3T because you now push the limit, you increase your resolution. Your increased signal to noise is kind of spent for having higher spatial resolution, and we run into the same problems to see these small effects, do the cross-validation, statistics, and everything. But it's really encouraging. For example, here's a slide of Elias group and a paper from Federico and Michel, where you basically see you can push it really forward and already see interesting feature representation, which I'm very interested in. Here in the auditory domain, you see tonotopic maps in very tiny structures, like here in the inferior colliculus. I think this is remarkable because the structures are so small and you still see a nice tonotopic gradient over sound frequencies. And this is really encouraging that we really are on the right track with this high-field imaging to resolve in local regions feature representations. Another study which Michel just presented at ISMM also shows that you can do the same thing in the medial geniculate body, and this is another nice result coming out of high-field imaging. And I want to do something very similar, but I want to focus on, like many of our groups do, on doing this kind of high-resolution work by specifically having the hypothesis, can we separate representations of information tangentially in the cortex within one specialized area, which is usually mapped as one area, like say the phase area. So now I'm not interested how active is the phase area. I want to see what features are coded within the phase area. What is the code used by the brain to code different phases? And that is actually the model we have in mind. And it kind of alludes to the question, what is the right level to understand the brain? And of course, if we have now the chance to get more fine-grained information into this question, then we hope that we can have better, deeper explanations of, in my case, as a psychologist, cognition, perception, how it works, how it relates to the brain. Can we make this so detailed that we can make models, neural net models, which kind of even kind of predict and kind of simulate brain activity data, like Brian has shown before, to predict a new experiment and see how much such a neural net model, which is driven by insights from previous work, integrated, how much it can predict the next data sets. That's exactly what we want to do in very similar logic. And when the classical approach, which I call, again, the macroscopic level, focuses mainly on this categorical level and on long-range connections and so on, the kind of mesoscopic approach zooms into the brain areas and tries to say, we know this area now, let's look what is coded inside that area. Let's look how connectivity emerges differently from different layers within this area and so on. These are the kind of questions we try now to approach. And the sad thing is, we can basically model perception and cognition to a quite satisfying event. Actually, I started with neural net modeling my PhD work, and I was really excited because I could have a model which shifts attention, which could group objects. So very cool, and it was even brain-inspired. But brain-inspired means basically nothing. It just meant you had two pathways and a visual thing, and that was it, right? So it's not. So you could have thousands of models which could do the same thing. So which one I should believe? And that was a bit also for me the driving force to go to high-field imaging, to fill these models with features which are basically more clearly coming from empirical data. For example, here's a nice model which can read invariantly text on a visual input. It's a really running model which does this. And it's working beautifully, but the features I use to code letters are just completely invented. I don't want to do this anymore. I want to have features which the brain uses, how the brain codes letters. This I want to put in this model to make it really deeply biologically plausible and not just akin to some abstract relationships. So that's actually the goal. And what do we have to do for that? In my opinion, to really put in this higher, not only in B1, but also in these higher areas which you can map with fMRI, to put within these areas features which are really kind of empirically derived to make these models much more realistic, then we really have to try to map these features. We have to find out what is coded in V4, what is coded in visual word from area, what are the features there. And this is exactly the challenge for the coming years, which I see myself working on as long as I can. And the other question is connectivity, not only saying there are these areas connected, but which features, say you have a lower level and you have a horizontal and a vertical feature, and now you have maybe a higher level area which responds only if these two features are exactly in this spatial relationship. So you might get like the detector of a T based on earlier areas which detect just horizontal vertical bars. It's very romantic thinking that is so easy, but it should at least drive us to try out how far we can come with that. So connectivity should also help us to build more complex features in higher levels of the cortex from features we find in lower levels of the cortex. And of course, the work where also Peter Manettini was involved, and especially, of course, it was developed by Nico Kriegeskort, who was a student at the time starting this work here and then moved on to Peter's lab. And it's actually, the searchlight was when Elia, me, and Nico discussed this here, was actually an idea to say, let's find areas and what they multivariately encode everywhere, anywhere in the brain. Let it just run around and let's not just look for this one voxel, but with the local neighborhood. And the idea was really the local neighborhood because I was interested, from my view on this topic, interested to find not kind of predictions using the whole brain, but interested to find, is it something like the information of a brain area and the next, and what information is coded to discriminate certain kind of presented objects to the subject. And that's actually one approach which is very successful, but of course you still don't know what's really coded in such areas. I'll give you one example of this technique later. So the idea is then to move on and say, let's try to solve this problem by increasing the spatial resolution and basically trying to go beyond this indirect black box approach of MBPA and directly try to map the features. If this would work, we would have a much closer link to electrophysiology, to kind of mesoscopic invasive technology. And this is, of course, very important to validate and learn from each other. It would also allow us to unravel feature codes in human cortex, and I will show you some examples in a moment. And very importantly, it would produce generative models, compositionality models. That means if I have a set of features and I really understand them, and now I see a new representation over these features, and I can combine these features weighted by their activity, I could understand really a new stimulus, not just those which are already learned. This is really a generative model if it really works. And this basically would also allow us to give us insights in differences in feature coding. So if I compare, say, dyslexic children with healthy controls and see some systematic changes in the way, for example, letters are coded in the visual word from area or letter strings, that could help us to gain new insights in such diseases. And finally, and this is of course not yet much done, but it's our goal to go in this direction, to also in learning studies, which Judith Peters and Joel and others in our group start now, to really look how these representations change with learning. So if you have faces you have never seen before, and now you learn them, can we see changes in feature representation in the face area during learning? These are questions we want to ask with this kind of approach. And I'm now going to the roots of this concept of cortical columns. Of course, you know Mountcastle started this in the somatosensory cortex, but I'm focusing more on the concept from Hubel and Wiesel in the visual cortex, and of course the electrical recordings that if you vertically penetrate, find neurons responding more to the same features than if you go tangentially. This kind of concept was even nicer brought up by these optical imaging techniques because you see over cortex more clearly how this kind of similarity and redundant or kind of coding for the same features, how much the extent is of this kind of sameness, which is defines roughly the borders of these columns. So this is actually very important work to go in this direction. And actually, the really crucial point is to understand that if we would have a brain where, say, in the face area, and it might still be there, we don't know it yet for this area, imagine that the feature detectors detecting some features of faces, like the distance of the eyes, the hairs, whatever, if the neurons would be randomly organized in this area of, say, 1 centimeter extent, and they would be randomly organized, I think then for my goals, high field imaging, for many other goals, yes, but not for my goals, would not be interesting. Because if you would increase the resolution from your 3T voxel size to your 7T voxel size, you would still get just an average of all kinds of features in your voxel, right? So therefore, it is crucial that we can base our thinking on the concept of functional clusters, which can be called cortical columns, because in the brain, it's not what we observe. We observe that the features coding, the neurons coding for similar features seem to be clustered, functionally clustered. However we call this, that they are functionally clustered is the key. If they would be just randomly displayed, we would basically have no chance to discover this with high-field imaging. Because there is a likelihood from animal research and from some human research that the brain really uses this, that neurons with similar response profiles functionally cluster, say in cortical columns, we have a chance to see it because the spatial extent is now no longer the neuron, but the cortical column which we have to resolve. And that is actually the key point. And you have seen this slide a few times now. This is, of course, a very seminal paper from Isa Yacoub and Camille's group, because it really showed for the first time convincingly in the human brain, at high-field imaging, combined with a spin echo sequence, which actually is very important, I'll come to this later, basically resolved and found orientation columns in V1. So that was really a key paper, which motivated me actually to write some grants going in this direction now as kind of a research program. And the problem with that study is that they were very smart. They selected subjects which had a straight calcarine. So they just used one spin echo slice, which was quite thick, but if you vertically average and the columnar concept is correct, it's fine because you average vertically across the column. But they couldn't handle curved cortex, so they had to select the subjects which had a straight calcarine. And the first task we set up in our lab was, let's extend this to curved cortex. So we developed tools which basically allow us to find these kind of informations also in areas where the cortex is no longer straight, which can be curved. Must not be much curved, but can be medial curved, can be stronger curved. And that was an important first methodological development we did. We also do not assume that columns extend literally the same vertically down. We know from work from Tanaka that this is not always the case. You often have more things like that columns are not very strict, that also other features sometimes innervate in some depths of columns. So the problem is, of course, much more difficult than the classical ice cube columnar model. We are aware of this. And therefore, we wanted to both measure or kind of model different depths layers and sample from different depth layers, and in the same time tangentially and in different depth layers. And this were the tools we developed. You see an example here. You see this is like a mesh, but the different colors mean the same mesh is kind of repeated several times and samples, depending on the local position, more or less in depth, depends on the thickness of the cortex. So it's an equal partitioning of the thickness of the cortex, so that we can sample independent of curvature and thickness, kind of where, with respect to the white matter, gray matter, and gray matter CSF boundary, where are we in the middle of this, and can selectively sample our data from these different cortical layers. Here are just three, because often three are enough. You want to have like more infragranular, granular, and supragranular layers, because that roughly distinguishes what we can see with our voxel size at the moment, and that's actually how we do it. And the important point is, if we have these kind of layer reconstructions, you see here, it's close to the boundary of the white gray matter. The blue one is very close to the boundary of the white matter CSF, and the green is in the middle. But you can use arbitrarily many of those mathematically. Whether your data has meaningful information and all of these, this is another question. But you can model this very easily now. And most importantly, for any of these grids, we call them 2D regular grids, which are not arbitrary spaced vertices of meshes, but they're really designed mathematically to really follow the cortex with a regular grid, so we can interpret them using metrics, comparing the different positions across depths and find correspondence and so on. So it's mathematically nice to use them. And we sample then, like you see here, from MP at these different depths levels, the functional information, and can then interpret that data, look how stable it is, and so on. And that's exactly what we do. But we also, like also Camille alluded this morning, and there's a lot of work done by Federico and Jan and Valentin, finding out kind of how is this dependence on depths for if we sample functional data from these different layers. And what we found and some others found, if you, of course, go to the higher, towards the CSF, the higher layers basically are driven if you use gradient echo, much more by the big veins, by the draining veins. And therefore, either you would not want to use them if you use gradient echo to not fall into this trap here. You would maybe then restrict it only to these layers. But if you're interested to look how the representations are across the layers, we need these layers too. And therefore, our solution is more to go with the gray sequence, which is a version of spin echo sequence originally developed by David Feinberg and changed over the years in Minneapolis and also here now by Valentin Kemper. So this sequence we use a lot and basically gives us kind of a more equally sensitive measure from all the different depth penetrations which we are interested in our reconstructions. And now I give you a few examples of this. Here is a motion experiment. You see, this was one of my first fMRI experiments, actually. And what we did at that time is we showed moving versus static dots. And basically, you see what also many others at that time did, like Roger Tuttle and others. You find that you get this many areas, but the most strongly responding one to motion and not to static stimuli is this area human MT+. And that's basically what we found at that time. And now with Jan Zimmermann, we had the idea, let's basically try to see whether we now can zoom in MT, we localize it with some classical localizers which are available with fMRI. And then if we have MT proper, let's look inside the voxels whether we can see whether these voxels are selective for the direction of motion we show. No longer just motion versus static, but do we get different responses if the dots move in this direction and when they move in this direction or in this direction. So that would mean we look now for the feature representations in MT. And that's exactly what we did, and it worked really quite nicely. And you see here, for example, our depth sampling. We get the functional data on here, in this case, we show 2 different depth layers here, and basically see now that there are regions in this MT area which respond to different directions of motion. Actually, we had not highly significant results if we went directly to 8 directions of motion. But if we group the directions of motion, opponent motions, like going this direction and this in one axis of motion, and these 2, and these 2, and these 2, then we get 4 axes of motion. And they were reliable, so we could cross-validate the data with other runs and basically could find very stable results so that we can map now these so-called axes of motion columns. And that works because the opponent directions of motion are in the brain located closely, so you basically get kind of like a bigger columnar region that's easier to find with 0.8 millimeter isovoxels, which we used in this experiment. And you see also that we get pretty nice tuning curves. That means the voxels respond mainly to one of these axes of motion, not to others. So it's almost always a kind of unimodal curve, and that's actually very nice. It resembles a lot what is described in the monkey MT area. And of course, this is just the begin. Just want to highlight here that we have to go forward now, having done this. And the simple thing on V1 and V5 is, even if it's very difficult to get this data, it's very challenging. But the point is, in these areas, we know the features. We know that V1, besides globally retinotopy, locally, for each patch of the visual field, we have a hypercolumn. We know the features there are orientation selective columns. And in MT, we knew it from Bill Newsom and many others, there are direction selective columns. So let's look for this, and we know what stimuli to use to probe the different features. So if we now go higher in the visual hierarchy or go to other modalities, we basically have to find out what features are really the ones which are the interesting ones to kind of partition the responses into meaningful sub-responses, which is very challenging. For example, we can go back to the strategy of Keiichi Tanaka and the monkey in IT cortex to kind of show objects and then simplify them. For example, if you have a T and you find a voxel in a visual word form area which responds nicely to a T, and then you break the T and you might find it still responds, then it doesn't care about this connection. But if it drops down in this response, then we would know this seems to be a voxel which likes to have these things combined in one new detected feature, right? So this is a very difficult and challenging work which we have started, but which takes a long time to do this for some of the areas we have in our list. And these are some of these areas. So we have worked a lot now on V5, bit on V1, now going also to the phase regions and the LOC regions and the visual word form area. So we now approach these other regions and we try to get similar feature representation results like for MT, but the result will be better because if it works, we will also know more what features are coded in these areas. Now I want to show you, coming back to Camille Camille's point this morning, where he said, ultimately, if these columnar concepts are meaningful, they should also be linked to behavior. And that's exactly what my original intention was. And you have to do one step before the other. So at the point you know the features in a certain area, then you can really use it in a predictive way. And what we tried to do is, going back to some work I did with Miguel Castelo-Branco many years ago, we basically used these so-called plate stimuli, and we reprogrammed them and did the study last summer in Minneapolis. And the idea is we used several mappings of the direction of motion columns first, but then the main experiment is now a different one, namely that we show to subjects these so-called plate stimuli. And if you look at them for some time, you might realize that you can see them as one object which goes upward, which we call pattern motion, and after some time you might see them as two sliding gratings, which they are by design. The stimuli are designed by two gratings which slide on top of each other. And if people look at this, it depends on the size and everything, so I'm not sure whether that works for you. But many people, if you do it right, perceive the same ambiguous stimulus either as this or as that interpretation. So one motion direction in the middle, or two motion directions going in this direction. And at that time, what we found was, with 3 Tesla, that the response in motion area is higher when people see component motion. So if you press a button and tell us what you see in the scanner, and then we look in the signal MT, when you see this kind of component motion, the signal is higher. But of course, we didn't know, is this because of attention? Like people like Higa argued, you might attend more because you like to see this more. We argued it could be that we have 2 assemblies of directions versus just one more active in these kind of switches. And that's exactly what we can now test, because we have the resolution of this direction columns, or axis of motion columns. And our prediction is then, if we have a constant ambiguous stimulus, but the subject's reports, I see kind of component motion, then the axis of motion columns for these kind of directions should be stronger. And if the people say we see now pattern motion, then the columns for patterns should be stronger. And that's what we set up in the experiment. And you see the voxels now here in this original GRACE data gets very tiny, and they're not smooth, nothing has been done to these voxels. And you can see if you average them in the localized MT area, we indeed replicate now with a GRACE sequence, not with gradient echo, we replicate the original finding in the paper we published some years ago. But the interesting point is now, if we let subjects press buttons in the ambiguous stimuli, we also find in the same MT area voxels which highly correlate with the state do I perceive component or pattern motion. And we can even predict this now from 2 runs to the 3rd run. So I did kind of some calculations and found that actually there are more columns for component motion which map mainly on mapped axis of motion for horizontal motion, and the red ones, which are smaller ones, map to the upward motion direction. And if I now use this data, then I see here in this original GRACE slice that indeed if the subject presses a button, I see component motion. The blue ones are more active. That's actually a validation result here. The blue ones go up, so to speak, and the pattern ones go a bit down, not to 0. They are still driven by moving stimulus, but they alternate in a very systematic way up and down. And this we relate now clearly to this axis of motion columns, and that helps us now to really see and link kind of the percept in mind to the level of the cortical columns. And I think that's for us very important. Another of these cognitive experiments is imagery. So we basically plan a lot of imagery experiments at the moment because we also want to test whether we can use internally generated brain activity for brain-computer interfaces, where we have a lot of research done in the last years, but it's all on categorical level. You imagine task A versus task B. This we can, like Peter showed this morning, also in real time, separate quite fast and nice. But we cannot necessarily separate, say, within a brain area that you imagine direction of motion to here versus direction of motion to there, at least not in a 3 Tesla scanner. So we put this at work with Thomas Emmerling and Jan and Zimmermann and others and tested that. Does this work on 7T? And actually we find it's pretty good. So if we have here a selected subject, and if we run a searchlight, which I showed before, over the data to discriminate which direction of motion, 1 of 4, we have kind of 4 different imagined dots move to the left, you have no stimulus, it's auditory cued. You imagine the dots move upward, completely without any stimulus. We can, with a searchlight, find 50 or higher %, 25 is chance level, accuracies here in the visual cortex, many areas actually, which can help us to discriminate what is the person, which direction of motion currently imagining. And in some subjects, this goes really to early areas, and might be that they have a topological attentional shift, whatever that picks up. We can also look in higher areas like MT proper and see whether it's possible there. And this we also did. We had a second ROI also in MT. And here you see the results, basically, which voxel's preference was by separating the imagined direction of motion. And you see there is indeed a topographic effect during imagery, which the classifier picks up. And you see here also the different subjects. So we have some which have very high accuracies, even going close to 100% for both kind of the early visual areas, but also when we just restrict the data to MT. In this subject, this works also just using the voxels there. But not all subjects, so we get quite also high variability across subjects, which could mean some can better imagine things. We don't know yet. We have to find out. This is work in progress. But it shows that at 7T, we can have also at the featural level of imagery separate these responses. And finally, I want to just point in some directions we currently investigate with Marian Schneider, Nikolaus Weisskopf, and many others here, Ala Trubruck and many others, to try also to use the kind of laminar level to go in the direction of effective connectivity, to kind of model connectivity from one area to the other. And in the first work in this direction, we basically used the PAF method, and Jan Zimmermann and Federico extended that to the laminar level. That means, like Brian Wendel's work with Serge Dumoulin, they invented this beautiful method of PAF, which is the modern way to do retinotopy. You get more information, works actually nicer. But using this now and push this on the laminar level, this was the task and challenge which, as usual, Jan met very nicely. And basically, it means that we now use our technique I presented before and now do kind of PAFs separately for different depths in the cortex, in V1 and V2 in our case here, the first application of this. So that means we get PAF fits at the laminar level from one area, from all the lamina to the next area and backward, so that we basically see this kind of approach. And you see that and actually one other extension of this model is it's model free. So it's not assuming a Gaussian like the original proposal, but it can find any shape of receptor field. It therefore has to run on big computers. We use a GPU computer for this. Otherwise, it would be not have to wait too long for this. And you see here examples where this works very well. We got significant PAF fits, like you see here, quite small, nice source regions feeding V2 from V1. And here you see also when it doesn't work, it's not significant, and you see it's more a random result coming out. And by pooling now the data from the layers of V1 to the layers of V2 and backward, we look now in connectivity, functional connectivity. And we see here that basically from V1 we get an input to V2, and that fits very well the reports from Van Essen and others, how it should be. So it is the first step in the direction of using now laminar separation of functional data to do specific laminar connectivity. And my final slide, Elia, is from Lars Mukli's work, which we collaborate with Federico and the crew in Minneapolis, basically where he started with an experiment where he showed pictures and then had a quadrant without showing any information there during testing here. So he trains the full pictures and then uses voxels only in V1, which do not respond because you spare out this quadrant. And nonetheless, in this PNS paper, he showed that if you use the voxels there, they have a high accuracy in predicting the whole scene you see. So you know one of the three which you really at the moment see, even if you use voxels, which are not directly stimulated because there is no stimulus. And the explanation is there might be some top-down effects. But where do these top-down effects end in V1? Of course, according to the general columnar and layer model, it should be in the superficial, supergranular layers. And that's what we tested. So we used, again, the same technique I showed you, this kind of depth modeling, separately extracted the data and found when we have the feed-forward approach, where the full picture is shown, the middle layers show the strongest predictive response because the quadrant is there. It's with information filled. The picture is full. But if I drop this case here, you see then the middle layers respond strongest. But if I kind of drop this quadrant here and use now voxels only here, which are not directly driven, then we see there's chance level in most layers except the superficial ones, which have a highly significant results, actually also across all subjects. So that shows that in principle we can now use this laminar separation also to apply, say, classifiers at the supergranular layers, middle layers, lower layers, and learn much more, is it more bottom-up, top-down effects, and so on. And with that, I want to conclude. Just mention there's a lot of more work from the auditory group and others, but I want to finish now and just want to summarize that with this kind of a new imaging center, we really want to push ultra-high field to go into this mesoscopic scale imaging, and this kind of imaging bridges these macro and mesoscopic scales, and that's we find very important. And if we can reveal these feature codes, this of course is important for neuroscience, because you understand maybe then better how information is coded also in the human brain, where you can look into this and have stimuli and cognitive tasks like attention, like imagery, and look at their mesoscopic responses. That's what we want to do. But the challenge is, of course, if we go beyond these clear areas where we know at least roughly what are the features coded in these areas and go to areas where this is much more unclear, that's a real challenge. It is not only that we need the scanners, we need also really to kind of understand and do clever experimentation. And I didn't talk about this, but this imagery work alludes to also our kind of brain-computer interface work. We also want to use more fine-grained features for doing this. And I want to end only by thanking many people here in this room. And I have to stress especially Jan Zimmermann, who was involved in most studies and did really excellent work. And especially also Federico, who was actually our hub between Minneapolis and Maastricht. And actually some of the experiments I showed you were done like he sends us a localizer of MT, Jan analyzed this and sent them the position back where MT is, and he did the gray scan in Minneapolis. So this was really fun, and I thank you all the others also for your contributions. Thank you very much.